Yeah, so the presentation today was basically to share a story that we recently finished, showing how important to look into gut bacterial metabolism, because metabolites of gut bacteria, this means that they are coming from processing food components, but also medication that we ingest. And this is happening all the time. But although we know that there is vast amount of these metabolites being produced all the time, we still know very little about them, but also about the metabolic process itself on the bacterial side. And what we showed today is this could be very important in terms of competition with the availability of the drugs. So in other words, effectiveness of the treatment in certain disorders. So today we focused on a bacterial enzyme that we showed to compete with the bioavailability of the main treatment of Parkinson's disease, that's the levodopa. And this enzyme is called bacterial tyrosine decarboxylase enzyme. So we as humans, we have an enzyme which is called dopa decarboxylase enzyme, and it's well known to break down levodopa into the metabolite dopamine. And that's why patients, when they are on the levodopa treatment, they are also given inhibitor of this human enzyme. Why? Because only levodopa can cross the blood-brain barrier to correct for the levels of dopamine there. But if it's already metabolized into dopamine in the periphery with this human enzyme, then the drug is not effective anymore because dopamine cannot pass the blood-brain barrier, right? So what we found out is that this inhibitor of the human dopa decarboxylase is not actually a potent inhibitor of the bacterial tyrosine decarboxylase. So it cannot prevent the bacterial enzyme from breaking down levodopa to dopamine. This means that if this gene is present in high abundance in population of Parkinson's disease, then there will be lower effectiveness of the treatment in there. And this is actually what we showed. Because during this study, so we did kinetic studies to characterize this bacterial enzyme. So we know like the strain specificity of this enzyme because its main substrate is actually not the levodopa, but it's the tyrosine, which is the precursor of levodopa. But when we incubated this purified enzyme with both substrates, the tyrosine and the levodopa, we found out that even when the tyrosine is in much higher concentrations compared to levodopa, it does not prevent the enzyme from converting levodopa to dopamine, only slows it down a bit in terms of the rate of conversion, right? But then the inhibitor does not work on this bacterial enzyme. So this is a major issue for those patients. And it's actually well known among Parkinson's disease patients that there is a high variability among the patients in terms of their use of dosage regime. So some of them require low frequency of dosage, like 3 tablets per day, but others require up to 11 tablets per day to get the same effect, which means they are also way higher susceptible to getting the side effects of the dosage, like, for example, dyskinesia. So together with our collaborators from Rush University, we took stool samples from patients who vary in terms of this frequency of the dosage of L-dopa. And then we looked into the relative abundance of the tyrosine decarboxylase gene in these samples. And we found out that the higher the relative abundance of the bacterial enzyme, the higher the dosage or the tablets that these patients were on. So there was a positive correlation in that case. What we also found is that there was a positive correlation between the abundance of this gene and the duration of the disease, meaning that the higher the abundance of the tyrosine decarboxylase gene, the longer the duration of the disease was in those samples that we tested. But something to mention there is that the main site of the absorption of levodopa is not actually in the colon, but higher up in the small intestine. So we might not be looking into the right sample, which is the stool samples. However, it was impossible for us to get jejunal samples or upper intestinal tract samples from those patients. So instead, we did an animal experiment. So we did rat experiments, normal rats, wild type, who have their, let's say, natural variation in terms of the microbiota that they have. And we gave them orally a mixture of levodopa and the inhibitor carbidopa. And then we took samples from their jejunal content, also from the plasma, so blood samples. And we looked into the levels of levodopa to see the availability in there. And we also measured the relative abundance of the tyrosine decarboxylase gene of the bacteria. And we confirmed what we saw in the patients. So the higher the relative abundance of the tyrosine decarboxylase, the lower the amounts that we measured of L-DOPA, both in the jejunal samples but also in the bloodstream. So this clearly showed that indeed the presence of the bacterial tyrosine decarboxylase would compromise for the levels of the levodopa available to reach the brain of those patients.
So, Sahar, can you tell us a little bit what you expect clinically from the results you just...
Yeah. So the clinical expectation from that is this work shows that the bacterial tyrosine decarboxylase might actually be used as a predictive biomarker in patients of Parkinson's disease on individual levels to already show who will respond or who will require higher frequency of the dosage regime from the beginning. Because actually from this work, we think that most of those patients, they start having, let's say, normal levels of these tyrosine decarboxylase encoding bacteria. But because we also know that Parkinson's patients, they usually have problems with gastric problems. And that's why they also take high doses of proton pump inhibitors. And proton pump inhibitors are known to end up in small intestinal bacterial overgrowth. And the dominant bacteria in there are actually the Enterococcus species, which are the main bacteria encoding the tyrosine decarboxylase. So we think that the problem starts from there. And then you end up with having higher levels of this enzyme, which will compete on the availability of levodopa. At the same time, this levodopa will be converted into dopamine in the gut. And dopamine is known to slow down the gut motility, meaning that there will be higher chance of constipation also in those patients. This is the first side. The other side, because it's like a vicious circle now, other side is that patients will start having higher frequency dosage of the levodopa. This means that the bacteria will continuously be exposed to the levodopa. And actually, these bacteria having tyrosine decarboxylase, we know from literature that they benefit from that. They can very well resist low pH, for example, and then they can overgrow. So then, again, you end up with a vicious circle there. So we really hope that's something we are working on now, to expand this to see whether we can use this bacterial gene as a predictive biomarkers in Parkinson's patients. Yeah, so our main goal first is to target the microbiota itself. So, for example, first we have to understand quite well how these bacteria utilize levodopa. Because if we understand this, then we can interfere with this pathway. That's from the beginning. Another option would be to, let's say, interfere with prebiotics or probiotics, because then they can take over the niche and then you don't end up with overgrowth of these tyrosine decarboxylase encoding bacteria. Yeah, this is the main way how we think we might solve it.
Yeah.
I guess the key messages from my talk are that we can no longer just study composition of the microbiota, what they're doing and what metabolites they're producing. This is what the host is being exposed to. It's a mechanism of information exchange between the microbiota and the host. There's the immune angle, of course, which composition is important for, but the metabolites as well play a key part in that. And studying composition alone, you don't quite see this exchange in that sense. Yeah, so we know that metabolites are produced in the gut and that these can be absorbed from the gut and then enter the bloodstream, into the systemic circulation. And once in the systemic circulation, they can reach peripheral tissues like the brain. And we actually know that there are receptors for some microbial metabolites in the brain. And so there are mechanisms set up for that crosstalk.
Maybe you can explain us why did you start to work on the brain-gut axis?
Yeah, sure. So there's a lot of, I guess one of the first motivations for studying the gut-brain axis was some initial findings that we had where we compared germ-free animals to conventional animals, and we looked, because we look at metabolites, we just looked at metabolites in the brain, and we found that they differed between germ-free and conventional animals. So it showed us that if you remove the microbiota from the mammalian system, you actually change the biochemistry of the brain. And that kind of came in parallel to some of the work that others in the field have been doing, looking at actual behavioural and functional changes in the behaviour. And so, that was the main motivation for looking further.
Okay. So you presented the American Gut Project as a crowdsourcing project. Could you tell us a little more about this project and how you came up to work with these samples and cohorts?
The way this study worked was that we would have the families samples at home, and it was kind of more interesting for a lot of reasons, but I guess the first one was that we had 15 states that participated. If you have a clinical center, then you're kind of constrained to one area, and so that could be a problem if you have so many different states and heterogeneity in your data. But the design of the study was families of 2 children, one with autism and one without, and they were all between 2 and 8 years old and within 2 years of each other. So we had a control groups that was moving along with the families. Mm-hmm. So that's how we try to counterbalance the problem of the heterogeneity of the populations we were working with.
What is it exactly that you've done then with this cohort?
So we asked them to answer a lot of questions online to start, and then we used a tool to try to confirm the diagnoses that were developed by Dennis Wall at Stanford, where they answered questions, and also we recorded a video. Mm-hmm. And he published all this work. But basically, we asked the families that they had an already diagnosed kids with autism and one that was not. And then with these two methods, we will confirm on top of the diagnosis that's supposed to already be done by a professional. And then after that, with those 2 tools, we would double check. So usually everyone had to answer the questionnaire, and I would say that 2/3 of the cohort sent us videos, and we would check between the correlations between the questionnaire and the videos and what the parents reported. And that way we could really only use the data for the analysis of the family that we knew would answer all the criteria we wanted.
Okay, and so basically the objective of your study was really to identify kind of biomarkers, correct?
Yeah.
Okay.
So there's been a lot of work on autism and microbiome, a lot. And there is quite a consensus about agreeing there is a dysbiosis in the microbiota in the kids with autism. And I would say there is a couple of fantastic papers. A couple of years ago, they did a fecal transplant, Kang et al. At Arizona State, is Rosa Cushman-Brown. Mm-hmm. And it was really, to me, striking on the microbiome is really something that seems to play an important role in autism. But there is not a consensus on really which community was involved, and can we find biomarkers, and can we test those biomarkers on mouse afterwards?
And so you have found this?
We found a few biomarkers. So we did as the best as we could in terms of analysis, and try to get the best control study that we could found, as I explained, with the family design. And we found a few biomarkers that we hope are something that actually is different between the 2 cohorts. And so what I did on top of this is I tried to take one of those bacteria, and that's the work that I do now at Oregon State, and I went into mouse and tested them to see if we saw behavior differences, and in one of the tests we see behavior differences. So I think that's pretty exciting.
So basically, what have been the pitfalls and barriers that you have encountered? Because I guess it's a quite difficult field of research.
Yeah, it is really, and the paper is in bioRxiv, so meaning it's open and people can read it, but we're still working on a Reviewverse to get it published in a journal. And it's not that easy to convince the reviewer for a few things. But I guess the biggest pitfall were, the clinical coordination, like having someone full-time recruiting families and, answering their question, they're worried, making sure what you do is legal. Like you can't have any of their health informations that is not protected, making sure you protect the people who participate. That their information is in a safe place, because you have their email address, their mailing address, some of their diagnosis. You need to be very careful, and it seems easy to put a website up, but you really want to make sure that the family will not be put in any, that their data are really safe with you. So that was putting everything in place, and Stanford and Deniswell was really key in doing this because they really have an entire setup for that.
And from this morning sessions, is there any a message that you really found of interest for your further work?
Yeah, I guess a lot of people have this consensus on the importance of short fatty acids, which we found as well. And then the gender differences, it's something that we noticed for a few of the work I'm doing in the lab now. And I noticed it with the data in the mouse, and I was not really believing in it because we're still working, and I didn't want to show this today or anything, but I was like, oh, interesting, because the preliminary data, we may see a couple of differences, but I honestly was like very skeptical about it, and we kind of put it on the side, and I was like, we'll see. But then this morning, a lot of people seems really into how the gender is different, like how, all the gender differences. So I'm interested to see where that field is going with this part. So that was what excited me the most, but that's very selfish just because I had some data that I was thinking of before. I'm very excited by the metabolomic work that's being done too, and because we're going to dive into that too. And I guess one of the things that we worked on the paper was to try to be reproducible and to really give the code out and make sure people understand what we did, which is something that people don't do enough because it takes a lot of time. And we've tried to put the code out to really have people to be able to use all the work we've done, and it's a lot of extra work that's not that rewarded by anything, pretty much publications, or because it's not required in all the journals, even though we really try to make sure that people could reuse it, and I think this is where we should trend now and put all the tools available to the scientific community. And it's difficult because it would take you an extra 2 months because you're sharing your code, so it would be like sharing your notes, Yep. So you want it to be clean and nice and everyone to understand. So you literally have to rewrite a lot of it, or make sure when you code in the first place, it's clean. And it's difficult because I work with students and they're learning, so they don't do that really well. So it add an extra layer that we really work on doing, and I hope people will just use it and it would be useful.
It will bring value to the research.
Yeah, hopefully.
Okay.
I hope so.
Okay. So what can you say about this conference?
For me, it's a gold mine because I always have to go to one talk in the big microbiome conference about gut-brain axis, and here it's just you sit, and this is a whole 2 days, and you really, if you listen carefully, you have a very good view of where the field's at now. And I really like, the keynote, Jane Foster is always really, really great to listen to, and so for me it's fantastic since my lab works on that.
So what is your biggest hope that the research can bring to the autistic children in 10 years from now?
If we could alleviate some of the symptoms, at least the GI symptoms, the gastrointestinal symptoms, and that would be a big achievement. So finding biomarkers, from this type of studies, and then do some testing, and why not formulate a probiotic or something, a therapeutic, that would be, to me, that would be what really would be a big achievement. And so this is a very, very long shot, but it starts by finding the biomarkers, you know, that there is no other way to start. So I'm hoping that this really would help the population by giving back to them. And, you know, also the families really were involved. We had a lot of exchange with them. The clinical coordinator, she has a lot of, it was Jenna Daniels, and she really worked on it a lot and had a lot of exchange with the families, and they were involved in this, and I really wish we can give back. That's my big hope.
Yeah. And so, just to finish, I found interesting your comment that based on this morning's presentation, you may want to look now, or maybe in a future work, on the window of between 0 and 2 years old. Yeah. So that's something that maybe is of interest for your future work?
It is, and it's complicated because after 2 years old, a lot of the parents have a hint if their kids has autism or not, and then, you know, they can go see a specialist and get diagnosed. But before 2 years old, there's a lot of milestones that, you know, the medical people will tell you, well, you know, there is these milestones at 2 years old. So before 2 years old, it's a little more complicated because you don't really have a firm diagnosis. Though, you know, the window of therapy is also between 0 and 2. There is a lot of literature on that. So that would be nice. But that would mean like working with some samples and wait to see if the diagnosis is confirmed. But that's also, that would be really complicated, I think. We could try to see families that have autism already before, you know, with a risk factor or this type of things. But thinking about it, but it may be complicated to implement. And so, yeah, we'll see.
Okay. What are the key messages from your talk?
Thanks. So I think the key takeaways from the talk are that you know, we've been looking at the gut microbiome and how it affects addiction-like properties of drugs in animal models, and we have some new data that show that if we knock down the microbiome, if we decrease the microbiome with antibiotics, it actually reduces the formation of addiction-like behaviors in animals. And in addition to this, it also changes the way genes are expressed in the brain in response to repeated injections of drugs of abuse. And so this really suggests that the gut microbiome is signaling to the brain in an important way to control both brain and behavior.
How do you think that really the metabolites from the microbiome can signal to the brain?
Yeah. So yeah, we're studying these metabolites that are known as short-chain fatty acids, and they're actually things that are made by bacteria when bacteria break down fibers. And there have been a number of other studies that have shown that these metabolites are absorbed from the intestines into the blood and can signal to the brain. And so we're not the first people to show that. We're the first to use them in this particular application. But it's something that's been established that these come from the gut and actually can affect brain function.
Are you planning to go for a clinical trial?
Yes, I would love to go for a clinical trial one day. So I'm a physician scientist. I'm a psychiatrist, and I see patients as well as do lab research. And so that's something that we're working pretty hard on now, is trying to find ways that we can refine the things that we found and make them relevant to human patients. And it's a huge clinical problem, and it's something that we would like to be able to make an intervention to help people with.
Are you planning to do some research on probiotics?
Yes, I think probiotics are a very interesting topic to us. You know, most of the work that we've been doing has been kind of the opposite of that. We've been using antibiotics to kind of knock down all of the bacteria that are there. But I think that's something we like to do, is to see if we can use probiotics to promote growth of the appropriate species that would reduce some of these drug craving behaviors. I think that would be tremendously beneficial. It would be one way we might go for a clinical trial.
Do you think that one day you can extrapolate your research to other kind of addiction?
Yeah, so it's definitely possible. So we actually, a few years ago, we published a study looking at how the microbiome affected behavioral response to cocaine. And so we've been looking at it with cocaine and other stimulants, as well as the opioids that I talked about today. And so I think there may be some generalizable features wherein the effects we see of the microbiome might have similar effects in different classes of substance use disorders.
How do you start in psychiatry and end in the microbiome?
It's It's a really good question. So, I've been interested in neuroscience for a long time. I have been working in neuroscience labs since 2003. My graduate work was more in like how proteins interact with each other at the synapse. And then when I came to my psychiatry residency, I started thinking about ways that I could come up with a project that would have more of a possibility for clinical translation, something that could actually go from the lab and then go to a clinical trial one day. And I read a bunch of work on the gut-brain axis, a lot from John Cryan's lab and others, and I thought it was really interesting, and I like the idea that you might be able to manipulate something in the gut and actually affect brain and behavior in ways that are important for psychiatry. And so it's that potential link to the clinic that I think really excited me about it and kind of started me doing this work.
What exactly do we know what the relationship is between our microbiome and addiction?
So there's actually not that much known about the relationship between microbiome and addiction. We published a study about 2 years ago looking at how depleting the microbiome affected response to cocaine. It actually enhanced the rewarding effects of cocaine in the short term. We've now found that depleting the microbiome reduces the rewarding effects of opioids like morphine and fentanyl. And there are a few other studies out there looking at how alcohol and the microbiome interact, but in general, it's a pretty new field that's really, it's starting to grow rapidly, but it's pretty new.
So you work on opioids, so it's like really specific. But for the alcohol addiction, it's difficult because the patient don't drink always the same alcohol.
Yeah, it's a really important point, and it actually can affect opioid research as well. But I think when you're doing these studies on patients who use alcohol, it can be affected by the kind of alcohol they drink. It can also be affected by the fact that very heavy alcohol users don't tend to maintain nutrition very well, and so that's something that can also affect the microbiome. And so it can be hard to parse out the effects that are due only to the alcohol versus due to changes in diet. There are some similar things with opioids. I mean, people use different kinds of opioids, which may have different effects on gut motility, and kind of some of the same issues that maintaining nutrition actually make it a little bit more difficult to figure out what changes are due to the drugs versus what's due to kind of the lifestyle that goes along with pathological substance use.
Mm-hmm. And are you planning to look at sex differences in your studies?
I'm definitely planning on looking at sex differences in our studies. We actually, my graduate student has a set of mice right now that are drinking their antibiotics, and we're going to be doing the same studies in female mice as well. Because I think it's, we saw some earlier talks today showing that there's a lot of male and female differences, and I think there are male and female differences in addiction as well. So women who use drugs are more likely to become addicted to them. So I think there are some potentially really important microbiome and sex interactions in drug abuse literature.
So Robert-Jan, could you explain what the key messages are from your talk you just gave?
The key message from my talk is that how food affects brain function is a very integrative thing, and it has to do with changes in microbiota, it has to do with changes in metabolism, it has to do with the liver, with the blood-brain barrier, with the intestinal barrier, and it is a very integrative and complicated process, but that there is a possibility to affect brain function positively by changing diet.
You also talked about mice and men. Can you tell a little bit?
Yeah, what I normally say is that I don't like to discuss the small steps for men, which are giant leaps for mouse kind. And the reason is that, especially in gut-brain interaction research, the mouse models or the animal models are not that very predictive for the human situation.
What do you think are the most promising directions considering gut-brain research, and maybe especially for your type of research?
I think that regarding what is the promising venue or the promising window for gut-brain research is, I think nowadays is especially around anti-inflammation. If we look at dietary interventions, anti-inflammatory, and also, of course, the personalization, the precision management in that. And then also the role of the intestinal barrier comes in, because that's an important factor of inflammatory stimulus. Mm-hmm. So I would summarize it by how food can affect brain function also by decreasing inflammation.
Okay, you just talked about the most promising directions. What are typical pitfalls you envision in this type of research?
Yeah, in the human research, of course, the big problem is the inter and intra-individual variation in effects. And that is, we still don't know who are the responders and the non-responders. And we look at very subtle effects of intervention in humans. And then we have to deal with this immense variation in, if you could to compare it to an animal situation where they all get the same food, where they're inbred animals are in the same cages and so on. So that is the big challenge for human nutritional research in relation to gut-brain interactions. Biomarkers are okay. But
really?
Yeah, I think that we have a lot of biomarkers, but I think the variation in response to biomarkers, that's the big problem.
What will you bring back from this event?
Yeah, I just arrived because the flight was cancelled, so I'm not sure what I will bring back, but probably a better network and an improved network, because that's what I perceived last time when the first Mind Microbes conference, that it was very good for networking. And I think I will be more on level of everything what is happening around the world on this topic. Yeah, if you look at what has changed in the last 3 decades, I think the big change is that when I started with gut-brain axis research in humans, and especially in patients, and looking at psychological or psychiatric comorbidity in patients with gastrointestinal diseases and how we could affect that by gastrointestinal intervention, or the other way around, how we could affect the gut by psychiatric interventions, I was regarded as a lost case. And they said I was a promising researcher, but this was just the wrong choice. And I think it showed that I'm still a lost case, but I mean, it shows that this research is still promising, actually. So there is the concept has changed. So I think that is the big thing. The other thing is that it is still very much driven by technology, like the microbiome, like we see now with metabolome. So, but that's everywhere in medical research, that very much technology drives the research, like more than sometimes what are the unmet needs.